I'll start off by welcoming everyone here, and I'm broadcasting today. Sounds like a news station from the traditional unceded territories of the Coast Salish peoples, including the Musqueam, Tsleil-Waututh, and Squamish peoples. So I'm acknowledging that. Today, we're very privileged to have Brandon Findlay talking, and he is working in Concordia, earned his BSc at Simon Fraser, and then a PhD at the University of Manitoba with Dr. Frank Schweitzer. And he has an Alberta Innovative Health Solutions postdoc fellowship at the University of Alberta with Dr. Bederas. You're currently an associate professor at Department of Chemistry and Biochemistry at Concordia, cross appointment to the Department of Biology, and he studies the intersection of bacteria and complex small molecules with particular focus on discovery and development of novel antibiotics and the evolution of antibiotic resistance. So obviously, very, very important area. Are you back from sabbatical yet, Brandon?
Technically not. I'm
Okay.
on campus at Concordia, but I have 17 more days of freedom.
So
Not that I'm counting or anything.
I can say that he's currently on sabbatical and recently spent time at the Public Health Lab at BC CDC studying genetics and evolution of, okay, carbapinamase producing organisms with Dr. Huang. Today, he's going to talk about antimicrobial resistance evolution from bench to bedside in BC hospitals. So welcome, Dr. Findlay. Appreciate you showing up and presenting to us. Over to you.
Awesome. Well, thank you, Michael, for the kind introduction. I will get this presentation going, provided, of course, that PowerPoint agrees with me. And so just let me know if you can't see. Everything appears to be going well on my end, but
Yep,
there's no guarantees in life.
it looks good to me.
Awesome. So I'm going to tell effectively two stories here. One starting with what my lab is doing at Concordia University, and then the other talking about what I was fortunate enough to be able to do at BC CDC. And I think there's some interesting parallels there, and hopefully you will as well. So just for the general introduction here, antibiotic resistance is a major global health threat. It has been declared a health threat by the World Health Organization, and it has been calculated to claim the lives of over 1 million people each year. This is something that's unfortunately getting worse every year, and this is the result of the infections caused by antibiotic resistant organisms being harder to treat, having fewer treatment options with worse side effect profiles. And so this leads to longer hospital stays and to higher mortality rates. It has been called a silent epidemic, which tends to bring us back to mind of plague, to bubonic plague, Yersinia pestis, and other flashier organisms. But this is a major threat that we're facing in the world today. And that is kind of what antibiotic resistance is to us. This is how it influences people, how it influences patients, the healthcare system. But what really got me interested in the work that I do is looking at it from the perspective of the bacteria. They aren't really cognizant of all of the damage that they do, but they definitely know when we start trying to treat infections with antibiotics. And I've really been fascinated by how they react and respond to what, from their perspective, is just a giant wave of poison that comes crashing down on them. This has been helpful both in terms of discovering new antibiotics, understanding kind of at the ecological level what antibiotics mean to cells and to the environment. And it's also been very helpful with the development of lab evolution systems. What I have here is just a little soft agar system that my lab developed and one that we use to study how resistance evolves and how cells move from susceptible to resistant. And the way this works is we call this soft agar gradient evolution, or SAGE. And what we do is simply pour one layer of agar with a suitable growth media into it, while a simple petri dish is propped up on one end. This causes the agar to solidify into a pretty little wedge. And then when we remove the strut and pour a second layer, we're able to get two triangular wedges. Here on the bottom left, I have one of the wedges filled with malachite green, just to give it a bit of a Gatorade feel, just to make it visible, the difference between the wedge that in most cases would have an antibiotic and the one that would be antibiotic free. So while we have this sharp delineation between the two, after about 12 to 20 hours worth of incubation, diffusion takes place, and we end up with a very smooth gradient, where from the top-down view, we have one end with little to no antibiotics, and then a far end that is at a much, much higher concentration. We then inoculate one side with susceptible bacteria, and as they grow, they start consuming resources, they start producing waste. And so through simple chemotaxis, they move their way from left to right. As they do, they swim into higher and higher gradients of antibiotic until this starts impairing their growth, it starts killing them, and growth stops. At that point, any cells in the population that are resistant will keep moving and leave behind all their brethren, either because they were in the population initially or because a mutational event has happened and resistance has developed. What this looks like in practice is what we have on the right here, where after 1 day of incubation, the cells inoculated at the bottom in this bright scar have moved their way up, they've stopped, they've formed a few nodules of higher density growth, and then over the next 3 days, from those, we get radiative growth of new, more resistant strains. This whole plate is roughly 10 centimeters or 4 inches long. And in general, we're able to get resistant cells to any antibiotic we choose in about 7 days or less. The really cool thing is, of course, we can see the population dynamics. We can see resistant cells. We can see the evolution of those lineages and their movement throughout the plate. And this is both really cool. It literally just looks really cool. But we can also stick a pipette into this, and we can sample at any point along this evolutionary tree. We can get cells at their peak when they've developed all the resistance mutations they need to conquer the plate. We can get them when they've got 1 mutation, 2, 3, however many it took for them to get to that point. And we can even look to see if they're getting compensatory mutations, if they're getting off-target mutations that just help them grow better. And so this is a really cool system. We've been working on this for a few years now, and we've really been trying to push evolution to its limits. We've been trying to figure out what the max antibiotic pressure that we can put on these cells until we hit a point where they cannot further adapt. The goal of this is, of course, to lead to better and better antibiotic treatments. And the story I want to focus on today is our attempt to build evolutionary traps. If we can't stop evolution, can we guide it in non-productive directions? Can we effectively guide bacteria to a point where their adaptive potential just fizzles out? And in theory, this would allow us to apply one antibiotic after another in effectively an infinite series. And this would allow us to effectively find treatments where we don't necessarily need to worry about resistance evolving, either because it's effectively impossible to do so, or because the cells have become so impaired through the development of all of these different resistance mechanisms that they're no longer a threat, they're no longer a going concern. And what brought us into this initially was the idea of collateral sensitivity. This is a phenomenon where as cells grow resistant to one antibiotic, their susceptibility to a second antibiotic increases. And so this was postulated to lead to cells that are hyper susceptible to the second antibiotic. And in the original interpretation, would cause them to effectively have increased difficulty evolving multi-drug resistance, or just evolving resistance to, in this case, antibiotic B. And this, I mean, I like the theory quite a bit, but when we brought this into the lab, we ran into a number of problems. So here I've got several different resistant lineages exposed to gentamicin, piperacillin, ciprofloxacin, polymyxin B, and tigecycline, respectively. And each of these, we evolved 15 or 16 replicates of the same lab strain. So we ran one evolution experiment, that became this little cell here. Then we went back to the start, ran it again with fresh cells to get our second, fresh cells our third, so on and so forth. And what we saw was we did see collateral sensitivity. That is the lighter orange yellow color or tan orange color here. But across all of these replicates, we also saw a number of cases where there was no change in susceptibility to the second drug listed at the bottom here. And in some cases where we saw both collateral sensitivity, no change, and even cross resistance. And so this seemed to show that evolution is stochastic. It does not take the same mutations, the same paths every time. And not every path leads to the same outcome, leads to the same level of collateral sensitivity or susceptibility to other antibiotics. The other thing which was quite disappointing to us was that even when we did see collateral sensitivity, the changes were honestly quite meager. We usually saw twofold changes to the second antibiotic, potentially fourfold at best. The difference between a susceptible and a resistant cell could be 16 to 256-fold change in the MIC. And so getting a twofold in collateral sensitivity did not seem like the sort of thing that was likely to be clinically relevant. What we did see, though, is when we took these resistant cells and then exposed them to their antibiotic Bs, they often did have trouble evolving resistance, evolving multi-drug resistance. Here, I've got single exposure. So when naive cells were shown gentamicin, in 16 out of 16 cases, we got resistance above clinical breakpoints. Likewise, when naive cells were shown piperacillin, 16 out of 16 times, we got clinical resistance. But when we took gentamicin cells and exposed them to piperacillin, only half of them were able to make it to the end of the plate within 7 days, our stand-in for extinction. If we gave those cells a second shot, the ones that failed, we gave them another go. In most cases, they were able to get to the end. So this definitely seems like a random outcome, but it does imply that multi-drug resistance is harder to evolve than single drug resistance for this strain of E. coli in SAGE systems. Somewhat surprisingly, though, collateral sensitivity did literally nothing. We got a p-value of 0 or greater than 0.9999, which I have never seen before. There was literally no correlation as to whether the cells were collaterally sensitive or not on whether or not they became extinct. Their prior path effectively had no impact as measured through collateral sensitivity on acquisition of resistance to antibiotic B. So what we did see, though, is in a number of cases, evolution of resistance to B impacted the susceptibility to that first antibiotic, to antibiotic A. So here we're tracking resistance to the first drug, so gentamicin in this one on the left, then piperacillin, then cipro, then polymyxin. And when they were evolved resistance to the second, in a lot of cases, the overall susceptibility to antibiotic A went up. So some of the piperacillin-resistant cells became piperacillin sensitive again, at least below breakpoint. Not many, 1 out of the 9 lineages here, but we were seeing a bit of a glimmer here that potentially, if we picked our antibiotics right, we could resensitize cells, we could effectively reverse the evolution of resistance. Polymyxin, for reasons that I won't go into today, proved to be incredibly effective here. Exposure to tigecycline completely reverted the cells below clinical breakpoints. Unfortunately, polymyxin also reverts kidneys into non-functioning organs, and so it's not used too much in the clinic. But the hope then is that we could take something similar and we can potentially use some more clinically relevant or some antibiotics with less potent side effects for a similar path. And so we then set out to say, how can we make this process as painful for the bacteria as possible? How can we make it so that every time they evolve resistance, it hits them where it hurts? It leads to ridiculous reductions in growth, ridiculous susceptibilities to other agents like other antibiotics. And we envisioned here 3 or more drugs that we could just infinitely loop and either completely wipe out the cells or continually restore susceptibility to previous agents in the loop. And so for this, we turn to nitrofurantoin and that gentamicin piperacillin pair. We picked nitrofurantoin because it had low levels of cross-resistance to the other two. It was clinically relevant. It's used quite frequently in urinary tract infections against E. coli. And it had a resistance mechanism that was orthogonal to both the aminoglycoside gentamicin and the beta-lactam piperacillin. In this case, through an activation of NSF-A and B, nitrofurantoin is a prodrug, and so these 2 enzymes in E. coli are required for it to function. Notably, nitrofurantoin has no consistent collateral sensitivity to either piperacillin or gentamicin. There's effectively no consistent blue whenever we're looking at it in the chart on the right. But given our 0.9999 p-value, collateral sensitivity didn't appear to be a significant concern at this point. So fortunate for us, and hopefully for others, when we brought nitrofurantoin into this loop, we were able to consistently restore susceptibility to gentamicin. Here, we're just tracking gentamicin MICs as cells evolve resistance to gentamicin, then piperacillin, and then nitrofurantoin. And while we do see some resensitization when piperacillin evolution occurs, there's much, much more when nitrofurantoin comes in. These cells effectively do not wish to be triply resistant to all 3 drugs. This also worked backwards when nitrofurantoin was the first and gentamicin was the third. Again, we're able to reliably reduce MICs against nitrofurantoin, in some cases below clinical breakpoints, even if only just. And the way this appears to be working is through stacking of all those fitness penalties. When we split the cells into those that lost their resistance to gentamicin that became resensitized, and those that held onto it, we see a clear separation in the bacterial fitness, here measured as just area under the curve, a value that combines both changes in lag time, changes in growth rate, and maximum ODs when growing in rich media. The fitness of the cells actually goes up when they become nitrofurantoin resistant if they lose gentamicin susceptibility, not by much, but by a little bit. But it takes a much more significant penalty if they try and retain all 3 resistances. And so I think this is just highlighting that resistance is not free in many, many cases. And depending on the environment, it's not even particularly adaptive. These cells had not seen gentamicin in a few weeks, and so that was enough for evolution to effectively say, this gentamicin resistance is not benefiting us. It's more of a liability than a benefit. And so causing causing the population to be biased towards gentamicin susceptible mutations. But this is, of course, with lab strains of E. coli, specifically E. coli BW25113. And much as I love it, it's not the toughest E. coli strain around. It's been in a test tube for decades, and it has very few ways of combating antibiotic pressure. So the real question then is, okay, it works on the bench. Will it work with something a little closer to what you might see in a clinic? And for this, we managed, Philippe Bilek from the Medical University of Lodz was kind enough to supply us with some clinical isolates of E. coli that were resistant to nitrofurantoin. We then split these with 8 replicates each and exposed them to piperacillin and then to gentamicin to try and replicate that earlier 3-drug series. Because these were clinical strains, they had seen an antibiotic recently, and so they generally contained beta-lactamases and had the ability to break down natively piperacillin. To stop that, we paired piperacillin with tazobactam, creating a piperacillin-tazobactam sage plate. And what we found here was that we were able to resensitize some of these lineages to nitrofurantoin. It wasn't consistent. It did not happen in every case, but we were able to drop MICs a chunk of the time. Here, the dashed lines correspond to what the initial MIC was for each of these different strains, and the triangles are then where it was after exposure to pip/tazo and to gentamicin. If the lineage went extinct, it got a cute little skull here. And so for A and B, we dropped the MIC 2 or 4-fold. For lineage C, there was no overall change in those that survived, but we did wipe out half of them. And for D, most of them survived, and they survived at the same MIC. So I don't believe this is a silver bullet yet, but it does suggest that by judicious selection of antibiotics, we can potentially guide evolution to destinations that are less beneficial for the bacteria and thus more beneficial for the people trying to combat the bacterial infections. Oh, and one animation, just showing them again. We did see in one case mutation to piperacillin through changes in the beta-lactamase, but that did not seem to matter too much here. We still got resensitization to nitrofurantoin. This is something we will have to look at further because this was more of a one-off than a consistent mutation in the beta-lactamase, but it does suggest that these mutations don't have to be purely based around efflux or changes to the cell envelope. All right, so just to summarize here, mutations come at a cost. Resistance comes at a cost. And so this is something that we can potentially use to control or guide evolution. By selecting things, we can effectively find situations where there are no good options for the bacteria. Even with compensatory mutations, they are still going to be taking consistent punishing blows to their fitness, which will render them either more likely to be resensitized, more likely to die, or more likely to be wiped out, hopefully by the human immune system. But resistance, unfortunately, in the clinic is not purely based around chromosomes. It's not purely based around effectively getting antibiotics into or out of the cell. There are a large number of inactivating enzymes, enzymes with the ability to break down 1 to 50 or more different antibiotics at once. And so I was fortunate enough to spend the winter in BC this year at BC CDC working with Dr. Linda Hong on carbapenemase producing organisms. And carbapennamases are rather broad spectrum enzymes that break down the core beta-lactam in penicillins, in carbapenems, in cephalosporins, in beta-lactam antibiotics. This allows them to nullify the best antibiotic classes we have, and in the single step can wipe out around half of the antibiotics that are used in the clinic. There are 5 major classes. I've got their protein structures shown at the bottom here, and they are steadily growing in prevalence both in BC, in Canada, and across the world. And this giant, beautiful heat map here are all of the carbapenemase producing organisms that have been found in BC and the plasmids that contain their carbapenems. Most of these are close to one-offs. We'll see a single infection at one point, and then nothing for weeks, and then perhaps we will see that clade, that carbapenemase containing plasmid again. What I want to focus on, though, are two different clades. One that carries the carbapenemase NDM1, which I'm going to call AA271 through this talk, and another which contains a KPC3, which I'll call 281 or AA281. The reason I want to focus on these is that they're both very prevalent. We see these more often than almost any other carbapenemase in this data set, and they're consistently found. This is not a case where we see it every blue moon, or every once in a blue moon. This is a case where these crop up week after week, year after year. And that, unfortunately, is because these carbapenemases aren't moving through BC. They're not associated with international travel. These ones live here. NDM1, or AA271, NDM1, is in the top left. And while there are a few cases where the patient had traveled outside of Canada in the last 12 months, the vast majority of these have no international travel association. Similarly for AA281. So these appear to be endemic. They are well settled in BC and spread across multiple different health authorities, multiple different geographic locales. Unfortunately, they're also both increasing in prevalence, and they are spreading more and more. So AA271 was first observed in 2011, and while its prevalence has not literally increased year over year, there are ebbs and flows, it is much, much more prevalent now than it was in the early 2010s. It is also spread from Health Authority B here to every single health authority in BC. 281 is a later arrival. It was first observed in 2015, but again, it is also spread and is now both more prevalent and more geographically distributed. So that's a bit of a problem. This is not the way we want resistance to go. And what I was interested in was finding out, what are these two plasmids? What do they contain? How are they so successful? And hopefully one day in the future, how can we combat them? How can we drive back down this prevalence and hopefully wipe them out? One of the things that jumps out rather early on was that these are not associated with any individual pathogen. This is a little word map made from the 700 or so different isolates of 2071 and 281. And almost no strain stands out here. E. coli and Klebsiella oxytoca are the most frequently found. But we see this in well over 24 different strains from across Enterobacteriales. These bacteria have been found in infections as the infectious organism. They've been found colonizing patients just on their elbows, in their guts, just associated with the patient, but not directly causing an infection. And they've been found in the broader environment. So the plasmid is the problem here. This is not a case where we have one pathogenic strain that we can target laser focused. This is a case of the pathogen being effectively just a host for these two plasmid groups as they move from cell to cell, from patient to patient. Focusing in on 271 for the moment, it of course carries NDM1. It has the NDM1 beta lactamase, and that confers effectively complete or near complete immunity to beta-lactam antibiotics. It also carries ARM-A, which similarly inactivates broad swaths of aminoglycosides, taking down in one blow two of the most useful antibiotic classes that we have. It was first isolated in 2009 in Hong Kong, and since then has been observed in multiple different countries, obviously including in Canada. And I think we can reasonably say that this plasmid is globally distributed and successful in a number of different jurisdictions. What also jumps out here is it is a very well-conserved plasmid. 33 out of the 69 sequences that have undergone long-read sequencing have two or fewer mutations separating them. On the right, I have just a phylogenetic tree showing the mass distances between plasmids. And you can see at the top, the first half of this is just a straight line because these plasmids have effectively no difference in sequence amongst themselves. This is really striking to me because it means that we're not seeing any mutations, even over a decade of infections, even over a decade of persisting in the environment. Coming from an academic lab, we can't grow cells overnight without worrying about introducing a SNP or two. And so having plasmids that just are not changing on year over year is really striking. There is some difference when we look a bit closer, but they tend to be differences in the large scale. So there are two transposons, one roughly 4 kilobases long, one roughly 6 kilobases long, that are repeatedly deleted. So we see them missing again and again in isolates separated year by year. So here we have one from 2012, then 2021 to 2023. These our deletions are repeatedly cropping up. And this is happening while we're also seeing plasmids with full length. This, therefore, is just a hallmark of a mix of positive and negative selection. The plasmids are repeatedly losing this DNA because it's harmful, it's reducing growth rates, it's impacting the fitness of the bacteria that carries it. But the population as a whole is retaining the sequence because it's providing a benefit. So we're going a little teeter-totter-like back and forth between populations where it's predominantly missing and populations where it's predominantly present. And this actually was not too much of a surprise because the first of these transposons contains ARM-A. And as useful as it is at combating aminoglycosides, ARM-A has a very clear fitness penalty. It methylates this one guanine right here, guanine 1405, which forms a key interaction with the third ring of most aminoglycosides, with the three-ringed ones, gentamicin, tobramycin, amikacin, so on and so forth. Without that interaction, these aminoglycosides cannot effectively bind. And so the MIC goes from single digits to 256 milligrams per liter or more. But the reason this works so well, and the reason the aminoglycosides target guanine 1405, is because it's essential to the actual functioning of the ribosome. And so methylating it actually introduces a little bit of sand into the function, into the gears of the ribosome, and that impairs in rich media growth by 13%, give or take, obviously depending a little bit on media, depending on the total genetic background of the cell. In natural environments, a growth penalty of 1 or 2% is backbreaking. So having growth reduced by 13% in many circumstances would render this completely non-viable. The reason RMA persists and the reason we see it year over year is because, well, it provides resistance to effectively all aminoglycosides that are used in the clinic. And so when we use aminoglycosides, we effectively counteract this fitness penalty, allowing cells with RMA to survive when their brethren die. The other transposon, though, was much more of a head scratcher. It has one aminoglycoside modifying enzyme, which provides resistance to a subset of aminoglycosides, AAC32D here. But as long as arm A is around, that's completely redundant. And none of the other genes in this cluster are well characterized. So this is a case where, like the RMA cluster, it's under positive and negative selection. But unlike that cluster, we don't know why. We don't know what in this cluster is providing such a strong fitness penalty. And we don't know why the plasmid is, well, why it's beneficial to have in that plasmid. Something is helpful, something is harmful, but we need to do some more research. We need to figure out what these genes are actually doing before we can figure out what those somethings are. Moving to the second one, this INC-N plasmid, similar to the 271, provides a resistance to an array of different antibiotics. We have the KPC3 at the top, the 12 o'clock position here, providing resistance to beta-lactams. We also have a number of aminoglycoside modifying enzymes, AAC6', ANT3 double prime, and then APH6 and 3 double prime. This also gets resistance to ciprofloxacin and other fluoroquinolones through QNRS1, and resistance to sulfa drugs through both SUL2 and DFA14. And just in case KPC3 is not enough, this also carries two further two further beta-lactamases, OXA9 and TEM1. From a genetic diversity standpoint, this isn't quite as conserved as 271. We do see a number of different groupings here on the phylogenetic tree. And so the neat thing, though, when we look across all of the plasmids that were sequenced through long reads here, we see at least 6 different transposons. We have TN4401B, which carries the KPC3, and then several others which I've named after their most distinctive genes. One was SUL2 and a few of the aminoglycoside modifying enzymes, and then several others with different resistance conferring elements. We also have 2 that don't have any clear function, one that seemed to be metabolic with FRM genes, and then one that I honestly found nothing of any consequence there, except for an addiction module that I've named Kupin. The reason that this clade seemed to be gaining and losing inserts so readily is because in the KPC3 transposon, there is a pair of genes, IST A and B. And this is a transposase and a transposase catalyst, which allows this plasmid to both gain and lose DNA very rapidly. By consequence, we also saw a huge number of inversions where sequences were just flipped around from the DNA almost coming out and then snapping back in. This is allowing the plasmid as its own entity to very rapidly change, to very rapidly effectively try out different gene cassettes like FRM, like SAL2, like the rest. But at least over the time period that we're looking at, over the period from 2015 to about 2024, 2023, there's no consistent gain or loss of transposons. We see one plasmid with almost none of the inserts in 2016, but then also in 2016, we see one plasmid with every single one of the inserts. And then from there forward, we see a mixture. Almost every permutation of having, say, transposon 1 and 2, but not 3, 1 and 4, but not 6. Almost every permutation is present in this data set. And so what this suggests is across BC, we actually have several different subpopulations of AA281, which vary principally in the transposon count that they have, in the various identities of each of these transposons. Evolution in flux, in other words. There isn't a stable, super effective sequence here like there is for AA271. But if we look a little deeper, there is still a lot of sequence similarity here. Just under half of the sequences in the dataset align with greater than 99.99% similarity, or fewer than 8 SNPs. I say greater than 99.99 because this is just a very crude blast, and at greater than 99, it flips over to 100%. So even while these plasmids are gaining and losing huge chunks of DNA, like the query cover here varies from 91 down to like 34%. Even while they're gaining and losing huge chunks, the actual sequence isn't changing. As with 271, we're not seeing a lot of mutations. We're not seeing a lot of insertions or deletions at the base pair level. So the overall plasmid sequence appears to be conserved, or at least guarded from genetic drift from random mutation events. Looking at a historical view here, there's actually clear antecedents for 281. It has high homology to 2 plasmids that were isolated from the northeastern United States, including plasmid 12, which was one of the first KPC3s to be found in the US. And it has also been found in Canada, specifically in Ontario and Quebec, where it was observed in 2011, roughly 4 years before it arrived in BC, according to our data set. This was picked up through the Canadian Nosocomial Infection Surveillance Program, and they also observed each of the transposons that are in BC, although they did not have any of their plasmids having every single transposon at once. It was much more of a jigsaw puzzle mismatch. And so what this is showing us is that plasmids can vary. Plasmids do gain and lose large chunks of DNA over time, but they appear to be pretty strongly conserved at the sequence level. And this was both surprising and not, because at Concordia, we actually had a plasmid mutation project that was unfortunately ended in the pilot stage for reasons that will become clear very shortly. So what we did is we took a plasmid that conveyed resistance to a beta-lactam antibiotic, put it into a naive bacterial cell, and tested its susceptibility. just to see, does this confer resistance? And then we attempted to evolve higher levels of resistance through SAGE. We took the plasmid out when cells reached the end of the plate and put it into a fresh cell, and then repeated our susceptibility testing. The idea being, if resistance was conferred through changes in the plasmid by separating the plasmid from the chromosome and then basically resetting the chromosome sequence, we would be able to successively evolve the plasmid and potentially predict how plasmids would change in the clinic. Unfortunately for us, while we were able to evolve increased susceptibility, reduced susceptibility, in this case to the cephalosporin cefazolin, every time we took the plasmid out and put it into fresh bacteria, we lost that susceptibility. The increase in resistance was always coming from changes in the chromosome, not changes in the plasmid. In this work, as it was in the surveillance, it's not clear why the sequence of the plasmid is so conserved, so privileged relative to the chromosome, but this does seem to suggest that plasmids do not change mutationally at the same rate that chromosomes do. So to bring this to a close, there are a large number of carbapenemase-producing organisms that appear in BC, but many of their plasmids are transient. They are little embers that come, are little sparks that come from outside the province, flare briefly, cause an infection, and then subside and appear to disappear from BC. At least 2 different clades are endemic, though. They are well integrated into the province and causing continued and ongoing outbreaks. These plasmids are gaining and losing genetic cassettes, which suggests that they are in flux. They're not stable. Evolution is continuing to refine their sequences. And unfortunately, that means they will likely become more effective over time, not less. This is largely changing through gross changes to the plasmid, through the addition of whole transposons with multiple genes. It's not coming through mutation in the plasmid itself, at least not on the timescales that we've been able to look at so far. And this low mutation rate in the plasmid itself is echoed in some early experiments that we've done in the lab, but it will take a bit more before we can pin down why things are the way that they are. So with that, I just need to thank the people who actually did all of the work. It's great that I'm able to present it, but for the vast majority of the time, I'm not the one to perform it. The early half of the work was done principally by Farhan Raman Chowdhury on the right here. The work at the very end was done by Prerna Singh. And in the middle, through my time at BC CDC, I have been highlighting work from Shannon Russell and Adriana Cabrera specifically, as well as a little from Hassan Hamsi. I'd also, of course, like to thank Dr. Huang, as well as everyone that I've had the privilege of working with, and you for taking the time to listen to my presentation. And I would, of course, be happy to answer any questions that you may have. Thank you.
Thanks very much, Dr. Findlay. That was an excellent, very interesting presentation. I'm a general pathologist, so it takes me back to my times of training in micro, and it's such a dynamic interaction. Very interesting to see this sort of evolution, and hopeful to hear that perhaps we can get some antibiotic treatments that are not going to develop resistance. Any questions for Brandon? Please feel free to put them in the chat, or if you just want to ask a question, if you can press your raise hand, because otherwise I won't be able to see you. About 40 people here. People are looking very shy, Brandon. Or everything was so crystal clear, it doesn't need.
I long to have one of those presentations. I'm not sure I've ever achieved it, though.
Dr. Hong is saying it was a great pleasure for us to have Brandon spark that enthusiasm in plasmid work. So there you go. ringing endorsement from Dr. Hong, Brandon.
Thank you.
Okay, well, seeing and hearing no questions, Brandon, again, pleasure listening to you and very interesting work. Best of luck in your career at Concordia, and I hope to see you visiting Vancouver soon.
You and me both. Thank you
Okay.
very much.
Bye, Brandon.
Thank you. Thank
Goodbye.
you.